And then it not only has to do that, but it has to change it in such a way that that protein is no longer functional. And that happens far fewer times than you would imagine. I mean, you can change something in an alpha helix, a number of different amino acids, and you still got a perfectly viable alpha helix. Okay. And if you happen to change one of those alpha helix amino acids out for a proline amino acid, all of a sudden you got a kink in the chain. It's highly deleterious. Negative selection is going to take it out. So, I mean, when you get down to the minutia of what's happening at the molecular level, genetic entropy is fucking nonsense. I'm sorry. It's just nonsense.
Is there any way... Snake seems to be knowledgeable on this. Would it be okay if we can at least let him in?
I can let him in. Well, do you understand what I'm saying about this problem of accumulating mutations? I'm trying to figure out how a mutation would accumulate.
Right. Well, in order for a mutation to accumulate, it has to be neutral.
Yeah.
It has to be like pretty much neutral. So it can accumulate by genetic drift. Oh, hey, Snake. And
hey, AJ.
Hello. Hey, how's my mic? I'm driving.
Sounds good, right?
Okay.
It's actually pretty good.
So I actually had a... Oh, sorry, go ahead.
I'm offended, Andrew. Were you saying that I will be off topic and Nestle will contribute, but I won't? Is that the implication?
No, that wasn't allowed. Just Nestle.
No, we were suspecting your connection with Dinosaur Adventureland.
Wasn't when people come in here they start getting Lady Diva all of a sudden they just get Are you saying that I would not contribute?
Are you suggesting? Are you suggesting that
sounds like what you mean?
Sounds like little penis.
So I actually had a question for you, Snake, if that's okay, since you seem to have studied this subject. So you said back mutations are a lot rarer than compensatory. Yeah, I cannot pronounce that. Compensatory mutations. Could you elaborate on that, perhaps?
Yeah, so like back mutation would be like you start off with the original sequence and then it mutates. Back mutation would be going back to the exact original sequence, but compensatory mutations, the sequence, it's rare for a sequence to mutate in the exact position that it mutated before. Right, so it can mutate in other parts along the sequence, but it's much more likely that that mutation will compensate for if that original mutation broke it in some way. Other mutations along the line can kind of fix that function, even though the original sequence is different. The function has been restored.
So
that's what I mean by compensatory.
Right. So would it be kind of accurate?
I think a more important factor to consider is like most of the mutations that happen every generation is just lost eventually. Like when you have kids, you only pass half of your DNA down to the next generation. Of course, it depends on how many kids you have, but still, eventually, at least most of them will eventually get lost because of this process of sexual recombination. But
yeah, either you lose that mutation or it actually gets just kind of lost in the shuffle where the effect is gone. Even though you still have that mutation, it doesn't have the same effect before because other mutations will interact with that mutation differently.
Right.
And I don't think that's what creationists understand. The context, a mutation doesn't have like an inherent quality.
Exactly. Well, that's another thing, Snake. So when I point out recombinations, Standing always says it's not recombining the individual nucleotides. It's only recombining or swapping the genes, the alleles. Does that kind of make any difference in your opinion?
I don't think so, no.
Well, it would be like one parent's likely to have a completely different set of alleles. Well, not completely, but
Right. Different
so
enough.
yeah, if you carry one mutation, someone else is not likely to carry that unless that's been spread throughout the entire population.
Right.
So if you have like at least two different kids, they're likely to have two different sets if the parents don't have the same allele.
Right.
And if it's that bad, one kid dies and the other survives.
Yeah.
I have a question. If you have identical twins, do they have the same like DNA sequence, or is it are there still a lot of differences?
Oh,
No,
okay,
same.
okay, there's still a few differences. Like in every cell division, I think the number is like 0. I forget the number exactly, but I think there is roughly a few mutations every cell division. No, not every cell division, but
Okay,
still,
there are different levels of identical twins depending on when the egg divides and how many cell divisions there have been.
Mhm.
So there can be more and more differences.
Yeah.
So they'll tell like they told my twins they're 80% identical.
Yeah, like I think the number is like 0.3 mutations per cell division, and of course, when the cell when the initial embryo divides, you have still a few mutations, and some of them are going to be inherited by the one twin and not by the other. But yeah.
Okay, but if you remember, like in 2004, I think when they cloned that sheep, I think it was named Dolly. I mean, that the clone would have this. Would that have the same exact
Yes.
sequence? Okay.
Probably not.
Oh, really?
Also, because
really?
As the sheep develops, it's undergoes also cell divisions, and that will also contribute some of the differences too. So yeah, like you are a mosaic. Like not every single almost even in your body right now, some of your cells have slightly different DNA than the others. Of course, not the difference is not very big, of course, but even if you count up the differences, there are still a few mutations between the different cells in your body. But yeah.
Okay, Speed, I'm starting to get tired of standing. He's starting to promote the. I mean, is that allowed here or?
I mean, the link is in the chat, so I mean, there's really no reason for him to just not.
Full discussion to see if Andrew addressed the challenges. So is he linking his video? I linked the one that was on Skyo's channel, and that's where the conversation is. So
I mean, Andrew's not going to go watch another YouTube video while he's in here.
But does standing
denying?
Does standing dispute the fact that sexual recombination means that most of the mutations that happen every generation will be eventually lost?
Yep.
Well, you don't even need sexual reproduction because there are asexual species that there's like a species of fish that are all female, and they're studying them because they're worried about there's another word for it in science, but genetic entropy for Standing's understanding. They're studying fish because they don't understand clearly how they don't die off because they don't have sexual reproduction.
Right.
But
they
found out. Yeah, Miller's Ratchet, exactly. Yeah, and they found out that compensatory mutations are the most likely reason for that.
Oh, really?
And there's a species of.
They have a mutation selection balance.
Yeah, they're not observing genetic degradation in these populations. So they're like, our models don't fully understand this yet. So our models say they should be dying off, but they're not dying off. So clearly there's something.
Do you have a link to something on that with compensatory?
Yeah, I can. Yeah, I'm in the car right now, so I can't link anything. But
I know there's also a species of salamander that's all female, and they do a mock courtship ritual to sort of initiate ovulation, and they give birth to, I think, clones of themselves that are always female.
Mm-hmm.
And
parthenogenesis.
Yeah, parthenogenesis. Yeah, something like that. And if this happens to humans, then
God created lots of lesbian animals for some reason.
Yeah, we have scissoring salamanders.
Yeah.
So so another question, Snake. In the context of the commonness of compensatory mutations, going back to back mutations, how rare would they be in comparison?
Let's
is
see.
there an intuitive way to think about that?
Back mutations are pretty rare. Let's see.
Yeah, they have to hit exactly the same
I know.
nucleotide and mutate it back.
Yeah. But I know, I mean, we do observe them, but I know that they've measured frequency of compensatory mutations up to 70%. And then there's research that says there's like up to 9 or 10 different solutions of compensatory mutations for each deleterious mutation. And the more deleterious the mutation is, the more likely it is to be compensated for by a different mutation.
Yeah, because it would not end up in the population.
If you break like a helix or something, just as a simple structure, and it's just like spinning around or something, if you change something else, it's likely to stabilize that structure just because you're changing things around. And the rest of the protein is, or the protein is mostly the same structure, so it continues on as it was. Most of the parts of proteins are not like actually functional; they're just like holding structure.
Right.
Functional, as in a functional group that chemically interacts with something else other than itself.
So here's another thing. Like, to me at least, this idea of effectively neutral mutations that are also slightly deleterious in their effects, isn't that contradictory?
Well, if they're effectively neutral, then it's contradictory. But usually what happens is they add up, and like certain mixtures of them will be deleterious. But on their own, they're neutral. And this is something I have talked to Standing to several times, and he never gives a good answer. He said, so it's like the effect of pleiotropy, where mutations will interact with each other. So you can have like different mixtures. Like if you have A, B, and C, that's fine. But if you have A, B, and D, slightly deleterious, each individually they're fine, and in some certain mixtures they're fine. But in a very specific mixture, they can be really bad.
Yeah.
And so that would be the kind of cure for that is epistasis, and that effectively makes these individually neutral mutations visible to selection because it's all of a sudden deleterious. And so I've asked Standing lots of times, because he'll say that can't solve anything because that just makes the problem worse, because if the combinations are making it more deleterious, that contributes more to genetic degradation. And so I asked him, and he also claims that it's not visible to selection. So I asked him how it can simultaneously be increasing the deleteriousness and be invisible to selection, and he never answers because they're completely contradictory statements.
Sounds about right.
So it is contradictory to say it's effectively neutral and that's accumulating, but it's still deleterious enough to cause a decline in fitness over many generations.
Yeah, it's like a word game because they're individually neutral, but together they're deleterious and therefore visible to selection. And you can select out certain combinations of these genes, but if nearly neutral mutation A is spread throughout the entire population, it doesn't matter because it is only deleterious in combination with certain others, but in combination with different ones, there are good and bad combinations. So
and that's kind of where recombination plays a role in kind of coupling all the bad ones together so that they amplify the deleterious effects, right? Kind of like what you're saying.
Yeah.
Well, I wrote something in the chat, and maybe Snake, you can say if this is true or not. But what I said was that as long as the percentage in a given population of beneficial mutations is more than 0, they're going to be selected for.
Mm-hmm. And the thing Sanford, I think, cites for his model and quote mines is, I think it's a Kimura paper that says that when you add in positive mutations, it makes the model unrealistic. But that's only because if you add in positive mutations, the rate of evolution goes up so fast, because even a small amount of just purely adaptive mutations, which are different than compensatory. Adaptive is like actual change to enzymes instead of fixing deleterious. Even a small amount of that just allows fitness to go up so like rocketously high that the model just allows for like really vigorous, fast fitness evolution. So it's actually the opposite of what creationists try to paint it as. So and we know there are beneficial mutations out there, and Stanford's models, he just claims, he just sets that number at 0 arbitrarily, and so that's a lie.
Yeah, that's one of the things that younger creationists say, because when they say beneficial mutations, they're talking about humans, but it's just not true to say that there are none, because we have recorded examples like bone density or resistance to AIDS or malaria. Like there's a lot of them.
And wouldn't it also be the
of course, they will point out, like in the malaria case, they will point out, like, oh, there is of course a cost associated with the mutation, but
that one they misrepresent that one.
Beneficial is context dependent always.
Right.
Like with antibiotic resistant bacteria, they say, well, it's devolving because while it evolves resistance to this antibiotic, it's actually losing fitness in the wild. But they have done studies where they put those bacteria back in wild conditions, and they observe compensatory mutations that restore their original fitness for that environment and retain their antibiotic resistance. And that occurs in the 75% of the populations. Some of them do accumulate mutations that are bad for them, and they die off, but that happens to be a minority.
Well, I mean, if you look at the number of species during the history of life on Earth that have died off, it's a lot of them. So, I mean, it makes sense.
You only need 1%, or even probably lower than that.
Yeah, I mean, and we've seen species go extinct in our lifetimes, or in past human history, like the dodo bird, etc.
Right. Well, I guess another thing to factor in might be effective population sizes on whether these slightly deleterious mutations can be acted upon by selection, right?
Oh yeah. It's the larger the population size, like the more effective the selection is. So you in large part, if you have a mutation that's slightly negative. In a very small population size, the effect of drift drowns out the effect of selection to select it out. But if you have a very large population size, then selection is more effective in selecting these slight deleterious mutations. Yeah. And
they've purposefully broken the genes on. There's this one study I know about. I'll link it to you guys in either chicken or some kind of reptile egg, I think. And they broke one of the genes on purpose, and then they watched what would happen. And the compensatory mutations restabilized that structure, even though it wasn't back mutations. So it wasn't the original sequence. And creationists have this idea that the gene that is there is the only possible combination of nucleotides that could give that gene that particular function, but there are thousands of different variations on an allele that can give you the exact same function.
Creationists think that the fitness landscape is consists of highly sharp peaks and nothing else. It's valleys and flat surfaces mostly. It's not just tiny peaks.
I have a
Because
question.
there are some genes that have they have a weak point where you can change one nucleotide and it'll all fall apart. But that's not the case with every part of that gene or every gene.
Okay,
it's often the case that like many proteins have like a core called the catalytic triad, where you have like 3 or maybe a few more amino acids that are really important in the catalytic process, but the rest of the mutations is mostly structural, and there's a lot of leeway when it comes to that.
Yeah.
Yeah.
And of course, the worse it is, the less likely to spread it is, which is why he has to go with the nearly neutral accumulation.
Right.
So Standing is in the chat talking about, how you refuted this and refuted that. What do you think he means by refute? Because when he refutes things,
well,
it means not refute.
He restates his original claim without actually responding.
He has several hours long circle jerk with other people, and just restating the same arguments over and over and over again, and then
and he seems to just show a paper that seems to kind of like say what he wants to say, and then he goes on to his next PowerPoint slide.
Yeah,
but I've never seen him actually drill down and refute something. But
you just restate your original claim using different words and with extra word salad, like extra, croutons and extra, seasoning.
Yeah. So I guess
I literally asked him, how can something be invisible to selection and increase in deleteriousness? And he'll just go back and state, well, it's neutral accumulation. And it's like,
right, exactly. It's that's why I was saying it's contradictory.
I understand that the initial mutations on their own are not deleterious enough for selection to be effective, but eventually, if you're arguing that eventually after many accumulations, then it will eventually reach a point where it becomes so deleterious, then it must have an effect of it or on selection. So there has to be a point where that happens.
But there are orders of magnitude times as many compensatory mutations that can slowly fix things while everything is accumulating too.
Yes.
Right.
And also there's also an important thing, like they often say that drift will cause the accumulation of the deleterious alleles, but drift will also cause the accumulation of the compensatory material. Of course, not in an equal amount, obviously, but still, the drift also has an influence on the compensatory mutations too.
I had a question for Snake.
Sorry. I'll finish this real quick and then.
Okay.
But there's a space of functionality where it can go a little better, a little worse, better, worse, and it can go up and down, but it's still functional within that space. It has to be really drastic to change function or to break function, which is,
Also, just to respond to Standing, like he said that I'm wrong because by that point the entire population is under genetic sickness. Like Stan has already responded to that by pointing out that according to the model, not every individual or not every ancestral line will accumulate these mutations at the same rate. So some individuals will reach the sickness point earlier than others. And by that point, you have a differential selection by definition.
Exactly. You basically have to posit that every individual has the exact same mutations.
Which I don't.
Right.
They all have different combinations of those mutations.
Yes, Stan has basically to argue that every single individual has the same, has the exact same mutational effect. It becomes very weird at that point. So it's basically a perfect accumulation at every individual, at every single generation throughout the entire population. Which is
that is correct.
Totally unreasonable.
That's correct, Stan. There are going to be some that are slightly more mutant. That means that they're going to be selected against.
Yes.
You just demonstrated the point. Anyway, what were we going to say, Peace Jr.?
Okay, so I think, it would be true to say that there are some genes that aren't alleles, but what would be the distinction between ones that are and aren't?
An allele is just some locus that has a difference in a population. So
right, it's just a differing version of the gene.
Yeah.
Like like how you have genes for different eye colors. And
Yes.
That's an allele. Okay, but what would be the I guess so you have a gene that I guess creates the iris in the first place, and then you have alleles that give the actual color. Is that right?
Well, it's a gene variant.
Okay.
So it's the same overall gene, but it has some differences.
Yeah, and like if you look across the human population, all of our genes have a tremendous amount of variance. And if you go look at you go look at NCBI, you'll find like 100 variants for some genes, or 1,000.
But not like most genes are pretty much identical.
Depending on how conserved they are. Okay, like if the gene is really identical across an entire population or across a number of different species, then you have a very highly conserved gene, and that one, if it mutates, is immediately going to be selected against, and that's why it's more conservative, right?
Right.
But there are some genes in which we have, like I think Dr. Dan said something about, was it insulin or one of them? There are like 1,000 alleles for that one gene.
Insulin? I don't know. I think
it might have been something else.
I think that's more typical with immune genes.
No, there are like if you look between if you look between a mouse and a human being and a chimp, right? The human being and the chimp will have like there's one gene. What the fuck is it? What is the famous one in chromosome 2? I can't think of it right now because I'm dead tired.
The fused chromosome.
Yeah, what is the pseudogene at that spot? I forget.
Oh, the DDX11L2.
Right. Okay. So DDX11 is a gene, right? And if you look at it from the mouse to the human and chimp are 94%, 92% or 96% identical. But if you look to the mouse, it's only 52% identical. That gene is not highly conserved. There can be a lot of variation in that one particular gene. Or if you look at the protein, you'll find that the amino acids vary by 52% between the mouse and the human, yet the gene does exactly the same thing. So,
well,
I mean,
you mentioned insulin speed, and we know that from Danny. Danny taught us that insulin is a mechanism.
Oh, it's a mechanism, right? That's right. Yeah, you're correct. You learned your lesson well from Danny. You should learn more genetics from Danny. And you can go join Standing for Truth.
I can learn about natural selection from Danny and from Fodor.
But no, the thing is,
I still don't understand that whole Fodor argument.
No one does. You just do a little better each time.
Oh, we're going to get into it again. We're going to invite Danny in the room because Danny, he won't come in the room with just me. He wants 2 or 3 others, and we could talk about that later backstage. But anyway, like. Okay, you've got an alpha helix here, right? Okay, there are a number of different amino acids that can fit into that, and that alpha helix doesn't change a bit. Okay, as long as they're like maybe they're hydrophobic or hydrophilic or whatever. But there are a lot of changes you can make to a protein, up to 70% in some cases, and still have functionality. So if you look across a population of human beings, which is, I mean, we are wide and mixed, right? We're a huge population, widely distributed across the earth. You're going to find a lot of variations. And that's kind of what they're mapping out now, is all these different variations. But I mean, protein function does not depend on an exact recipe. It's more like a 50% recipe, or in most cases, I think on average, you'll find 70% of the amino acids are conserved or similar. But you can look at the NCBI database, and they show you the conserved portions. There's a track that you can get that'll show you what's conserved and what's not.
Right. There's a lot interesting about like the immune genes that I mentioned before, like.
Oh, those things.
They are so diverse. Like we have many different versions of these genes, and interestingly, some of those versions are more similar to some versions in the chimpanzees than they are to other versions in our population. So it's a shared polymorphism.
Yeah.
Well, the immune system is really interesting, and I don't know if you guys know that channel in a nutshell, or Kyrgyzstan. How do you pronounce that? You know what I'm talking about? Like, it's an animated channel on YouTube with the little birds. Yeah, they started a new video series about the immune system that's going to be 3 parts, and I watched the first video, and it's really good. Obviously, all of their videos are great.
Can you throw a link in private chat so we can throw it out in the description so we can come back to this?
Yeah, but only if you link my new video in private chat in the description, because I put the link in there.
Oh God, you're always,
you're gonna make Speed irritable.
Well, I'm sorry. I'm coming out with so much new relevant content that I'm sorry if Speed can't keep up.
You can't throw a lot of content at him. He gets a little cranky.
Yeah, but I linked my new video where I'm talking. I did an interview with Hemant Mehta, the friendly atheist, and he gave some really great answers to my questions. So I recommend you guys watch that video because I'm shilling my channel. So
yeah, it's a good interview, and go over to Atheist Junior's channel and subscribe to it. And don't forget, Jason Torn on Saturday at 7 p.m. has a new channel. If you look in the description, there's a link to it. He'll be debuting with a discussion with Otangelo.
And do any of you have any like used cars you're selling or antiques or like a garage sale you want to advertise here? Because
in fact, we could just have people come in like on all those old radio shows, and announce things you're selling.
I'll put it in.
I just want to advertise me and Atheist Junior's epic rap battle.
Yes.
That is.
Okay.
I think it's still ongoing. I think you're gonna release your second one, right?
Oh no, I wasn't aware of that, but
I thought you said something like that. But...
What is this second link?
I already won, so.
I see how it is.
Okay, it says Speed. Can you link my new video in the description? What is the video?
It's the link that I put in that same exact comment.
I have the link, but what the hell is the video? I mean,
I wrote the title of the video in the private chat.
Is it you cleaning your sink or?
Well, no, it isn't. It's me talking with a friendly atheist.
Okay, you put it down below. Thank you.
And if I can just mention, if I can just show my channel one more time, I did a video about Kent Hovind getting arrested, and it got 1,000 views in like 4 days. Just saying.
Pretty cool.
Yeah. Who are you again?
I just noticed a comment made by Standing. Even with fitness variation in populations, you still have a population of mutants with some slightly more mutant than others. So he basically admits there is a fitness variation.
Yes,
so
that's what I was saying.
What does that mean? What does that mean? There is selection.
Yes.
Yeah.
So that's
AJ.
another.
Got them all linked.
Okay, I'm done derailing. You guys can get back on topic now. I'm sorry.
It looks to me like while our panel is talking about mutations and how they move evolution along, I think what's being put forth by our YEC interlocutors in the side chat is that mutation means we're all diseased.
Yeah.
And sin is just destroying everything, which is a totally different way of looking at things.
Yeah, well, what we got to remember is that they get all of their genetics starts in the Bible and ends in the Bible, and so they have this idea of this perfect genome, but yet they talk about all this mutation. They want 100% functionality, yet they talk about accumulating mutations. So they have to believe that their children are mutant compared to them, that they're less likely to reproduce than they are, right? Because if the entire genome is 100% functional and their kids are more mutant and they've lost information, then obviously their kids are less than them, right?
It's a very big problem. Like if your entire genome is like predominantly functional, then you have a very high genetic load. And by that, that means basically that any mutation will be very severe.
But think about that. Think about that in terms of cell division from the time you are a zygote until the time you are even a 3-year-old, a toddler, right? How many cell divisions you have. So if the genome were 100% functional and mutation is a thing, and we know that somatic mutation is a thing, there's no way that you could survive until you were even 2 weeks old. You would die of mutational overload.
Well, your body is kind of a natural selector because those cells will kill themselves.
Yeah, right. Some of them will
mutate.
Yeah. Of course, sometimes it goes wrong and you have cancer. Yeah.
Well,
but there would be only so many cell deaths before the embryo quit developing and failed to fix. And I think that is the fate of most mutations. Most mutations, 50% of the embryos that are fertilized never make it to pregnancy. So you never even notice that's happening. You've got a high natural selection at that level, at the level of fertilization and embryogenesis.
Well, even if you were to grant that the genome is 100% like useful, we know that some of the sequences in are just copies of copies. So surely those are redundant and could be removed. So I that's just one example of how seems to me like that's not very well designed if you've got redundancies in there.
Well, God's creation is so beautiful and wonderful, but also decayed and sinful.
Yeah.
It's also the case, like with, for example, with sperm cells, the ones that are more healthier, the ones that can reach the egg tend to have less mutations too, because if there's any mutation that causes them to be like broken or something, then they won't make it.
And I'm assuming this is for just the humans, but it also, like on the creationist side, the idea I think is that there's this ideal template of a human form and human body and biology that we're not conforming to because of sin, and that it's just getting worse and worse. But in actuality, there's no template. It's just that we had organic chemicals responding to the environment, which changed, and then the organic chemistry or organic life that came from all of that changed, and there's just been this constant push and pull between the environment and between creatures changing over generations, and two totally different mindsets.
So before Danny comes in, I had one last question for Snake, if that's okay.
Yeah.
So Snake, what are your thoughts on? Because Standing seems to confuse extinction vortex with air catastrophe, where there are some small populations where genetic drift causes them to go extinct, whereas there are other populations that are larger where selection is at play, and yet he still claims those larger populations are undergoing genetic meltdown, basically. What are your thoughts on that kind of equivocation?
Well, there is a kind of a limit. Like, so they say under about 100 reproductive individuals, or realistically more around 1,000, you'll start to see genetic decay.
Okay.
That's also what happened to the cheetah, I think, right? The cheetah reduced it down to like a few hundred individuals at some point, and now they are basically clones of each other, even though they are sexually reproducing.
It becomes pretty incestuous.
Yeah.
Which is a problem for the flood arc model, definitely, but not a problem for most animal populations.
Yeah. Well, because Standing, for example, pointed out this example of woolly mammoths going genetic meltdown, basically.
Yeah, Wrangel Island, yeah. This is a common example, but yeah.
And that's not the same thing, right? No. That's a different mechanism.
Yeah, as Stan Cardinal points out, these are direct opposite. Like in the mammoth example, you have lower genetic diversity because of inbreeding. Like you increase the heterozygosity of that, and you get recessive diseases most likely from that. But
it would actually be homozygosity, right?
Yeah, homozygosity. Yeah, sorry, I misspoke perhaps. But
okay.
Yeah, but with the mutation meltdown, like the genetic entropy, you increase the heterozygosity by getting new mutations that are deleterious. So they are exactly the opposite processes.
Also, I want to say that the perfect human specimens, they do exist, and clearly this proves creationism. And she's called Natalie Portman.
Yeah.
just want to add that.
I can't argue with you.
My spies tell me that AJ was talking shit.
I wasn't talking shit.
Oh, you didn't mention me? I guess
no. I mentioned you. I was just referring to you talking about. Oh gosh.
Where'd it go?
Saying that insulin was
insulin was a mechanism.
I wasn't talking shit though. Yeah. I like you, Danny. That's probably because you're one of my patrons and you bought me off, so I can be bought off. I'm basically a sellout.
AJ, he's not in the room anymore. Where'd he go?
I don't know where he went. I have no idea where he went.
Did you kick him, Speed?
Speed?
What? I'm not up to anything. I'm not doing anything.
Was I talking shit?
I don't know.
I was just doing some light ribbing.
Oh, what does Danny mean by insulin as a mechanism?
Wait, I just got that. So you don't think it's a mechanism then?
Insulin? No, it's not a mechanism.
Well, I got that information. I got that information from the Wikipedia.
Oh,
that is the primary.
Hold on. What do you mean by mechanism then?
Well, mechanism can mean two different things. It usually means either means something like a system of parts that work together to perform a function, or it can mean some kind of process in which involves those sorts of systems, right? And so in that sense, it's the primary mechanism. Insulin, there is a primary mechanism for blood regulation with respect to a particular kind of regulation going, I guess, downward, and that is insulin. It's perfectly sensible to say that. Biologists,
Danny, it's okay. It's okay to be ignorant. Okay.
Am I wrong? So you're just saying I'm wrong?
Oh, yeah. Like by the definition that he uses, I don't have a problem with calling insulin a mechanism. Like with the definition that he uses from that.
Well, I'm just using it the way that most people use it, and I think the wiki describes it.
Nobody uses it that way.
Well, look, this is the primary mechanism for release of insulin. Now, is that the insulin?
Well, that's the mechanism for the release of it, but not that.
That's what I read. The secretion of insulin and glucagon into the blood in response to the blood glucose concentration is the primary mechanism. Now, that's in line with the definition I gave, right?
You misunderstood it. You called insulin a mechanism, and you misunderstood it. And it didn't say that insulin is part of a mechanism. It's one of the elements or one of the mechanical elements.
I would caution saying that the insulin itself is the mechanism of control.
Right.
Of course,
it fits the first sense of the word. It fits the first sense of the word, right?
It's part of a system. It's part of a
nobody
system.
Would talk that
is the
way.
mechanism. Yeah.
Well,
the secretion of read this.
Does it fit the first sense of the word that I gave?
Up and read this.
I already read. I'm the one that brought you to that information, right?
Yeah, and you brought it like with your childlike understanding of science.
Wait, do you look? Slow down. Look, I'm the one that brought this to you.
The secretion of insulin and glucagon into the blood in response to the blood glucose concentration is the primary mechanism. The secretion is the mechanism. The insulin is not the mechanism.
I would say
that's what we were calling you out on.
I would say that the secretion of that hormone in the blood, and of course all the responses associated is the mechanism.
Yeah, the secretion. It says it right here.
Obviously, insulin doesn't work in a vacuum, but yeah.
Yeah, the secretion, but not insulin itself. And in fairness, Danny, if this is what he meant, then I would say that I agree with this.
Yeah.
But I also just love the idea of Danny complaining about somebody talking shit because he's one of the most adversarial people I've ever encountered on this platform.
Yeah, and he's in a mood today, so we don't need this shit.
I don't really have a problem with like saying that insulin is a mechanism, except for a bit of a reductionist.
Nobody would talk that way. Nobody would talk that way. It'd be like saying
I don't have much of a problem with it anyway. So yeah.
You'd really have to see the way he said it in the original context when we were talking about natural selection and how we said that natural selection is a mechanism, and Danny disagreed, and it was a roundabout discussion.
Yeah. Anyway, that was an aside and has nothing to do with anything. So, okay, back to genetic entropy.
Okay. Do we get Danny back or?
No, I don't want him back. He's in a mood today. I don't really care for him. So, is there some reason why is Danny going to add to the discussion on genetic entropy when he calls insulin a mechanism? No, I'm sorry.
Oh, sorry.
Yeah, it's not good to come into one of these type of streams when you have a bone to pick with one of the panel members, because that just sort of derails everything.
Kind of what's going on here, you know. I just don't see the need for it. What is
this thing that you keep sharing, Speed? Is this your child's scribbling from kindergarten?
Yes.
Oh.
Well, I hope they got a gold star because it's very, very well done.
Oh yeah.
Yeah. This is Danny. Okay, now you see him. Now you don't.
Speed's drawing is top notch.
It's top notch. Okay, this is a fucking gene. Okay. So the blue parts are the exons, okay? And actually I kind of misdrew it because God, it would be more like the blue parts would be a lot tinier, right? Like most of what you get in a gene is intron. Okay, and so the blue parts are the exons, and I made them bigger, okay? And the little green part before is the promoter. And the little red stuff that's way, way, way upstream is basically enhancers, regulatory enhancers that can either turn the gene off or on, right? So in order to make a change,
that looks like pink.
Are you being fucking Danny? Are you being pedantic? I threw him out. Anyway, okay, yeah, pink. Okay, fine, it's pink. So pink is promoter. Okay, so in order to make a change in this gene, you're going to have to hit one of those blue parts, or you're going to have to hit one of those white dots in the intron. Now, that isn't the whole story. Genomes are complex. In some of these introns, if you read it the other way, you'll end up with a few RNAs that end up having regulatory effects. But let's just not worry about that for a second. So you would have to modify something in the blue part. Now, if you modify something in the blue part, you're going to end up affecting maybe this alpha helix, and chances are you aren't going to change that alpha helix, okay? You might change something here, which is kind of the active motif of the gene, the part that actually does the enzymatic action. Or, if you modify enough in one of these alpha helices or in these beta sheets, you may change the effect of the enzyme a little bit so that it doesn't fit quite as well or something like that. But I mean, it's sort of, it's not exactly a Lego set, but kind of a Lego set. It's just that you can make a lot of changes before you're going to make a difference. Okay, so you have to accumulate a difference. Now, one way that it could happen is if you made like one nucleotide changes in this regulatory promoter. Now that promoter has to have something that attaches to it up here, basically. And if you keep making changes and it veers away from what that thing likes to attach to, it may attach less. And that means you're going to get less of that gene in this pathway right here, right? So you got this pathway here and it becomes mutated and you have less of this product in your cell, okay? Now the chances are that there's a compensatory mechanism that will turn on when this becomes low, this will turn on and it'll replace it. Okay, but if something happens to both of those mechanisms, like it's almost magical thinking that something would happen to both of those mechanisms and this product turns off completely. So now we have an accumulation of mutations. So that would be like they say that, the addition of mutations, right? That would basically add up to a problem. So all of a sudden, this thing, because of one more mutation right here, would end up killing the organism. and it would be selected against. So that's kind of what happens. Now I don't understand genetic entropy with this slowly degrading, like rust on a car, of an organism, because it's more binary than that. If I knock out this gene and that gene and that gene, I got a dead organism. Okay, so that is my problem with genetic entropy. Does that make sense?
Yes.
Crickets.
Chirp, 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 chirp.
Okay, does that make sense? I mean, I'm trying to steel man genetic entropy to think of a way. I'm trying to think of a way that it could, like, accumulate and slowly melt a entire population, like the size of humans, down to the point where, what would it be? You know, so maybe all humans end up having just this one mechanism, and then suddenly another mutation happens and starts knocking that out. Well, those people are going to die and they're not going to reproduce. And the people that don't have this are still going to be mutating. So what might happen is these little alternate mechanisms, okay, that aren't working, may develop a mutation that ends up creating that product. And then we have evolutionary change. And what's going on is that we probably have 5 different pathways that could make that product, and 2 of them don't work yet. Okay, but these are like pseudogenes. Okay, and for enough mutations, like in part of that population, this pseudogene may develop a pathway where it works for maybe just a small part of the population. So now suppose we knock this one out, knock that one out, knock that one out, knock that one out. These guys are going to be the population. They are going to reproduce, end up in the future, and that's how evolution happens. And it's that clear.
It's kind of a danger in if there's no more selection left, and increasingly there's less and less selection on human beings. But
right,
that's a change in environment. So if we have all these genetic diseases, but we can treat them with drugs and medical interventions, then all of a sudden the environment is making it so that they're not as deleterious as changes. So it's probably not as big a problem as. And the other thing is, if we lose the ability to a certain nutrient, like we can't produce all of the amino acids, but plants can. I don't know why God created us to do that. Well, we can just eat other things that have it in them.
Right.
And then we don't.
Well, the danger.
In our bodies anyway.
Okay, the danger would be that if this scenario happened and the entire population just happened to lose all of these pathways, and we were reliant on this one little tiny subsection of the population, like there's say there's 25 people in Cuba, and then a hurricane kills them all, and now we have an extinct species. We know species go extinct, and we know that it's because they end up in these loopholes, which are rare, but they happen, I guess, quite a bit. You know, there's a lot of extinct species. I mean, this is just evolution in action. That's kind of what they're talking about. So I don't see the problem. But I mean, if you look at this at the molecular level, there are a lot of questions for genetic entropy. And that's kind of what I'm pointing out here. What is the addition or what is the accumulation of mutations? What does it mean? What does it mean for an organism and for a population? So anyway, I'll shut the fuck up. Fuck you all.
Well, and again, you're also accumulating good mutations.
Yeah. Other possible pathways. And the thing is, we have all these pseudogenes, all these duplications, all of these transposons moving around, constantly shuffling around, creating new genetic pathways. And that's something that we've just barely scraped the surface on. We're looking for pathways that kill us, okay? And in that search, we're going to start to find all of these half-done pathways that are almost there, but for one mutation, they could be a functional pathway to create an amino acid, to regulate something or whatever. And it is such a complex, the body of different cell types, and that's kind of what we're doing with that story of evolution. Is I'm going to try to develop these ideas going forward so that you can visualize how evolution is possible. So,
well, what they don't realize too is that nearly neutral accumulation is actually, you could be building a really functional gene in the background the whole time.
yes,
And then
that's what I mean.
And then one last mutation kind of kicks it on.
Yeah,
And what they're complaining about is actually a huge mechanism for positive. In their mind, there's a positive direction of evolution, but fitness increase.
Yep.
Can I ask a random question?
No.
Yeah.
Okay.
If it's related to science and genetics, I doubt it's random.
Oh, well, it isn't actually related to genetics. Just has anyone seen the channel Science Asylum by Nick Lucid? So it's on.
No, negative.
Yeah, he just posted a video this morning about why we know dark matter exists. Anyway, just throwing that out there.
I have seen the channel.
What's it
Science
called?
Asylum?
Yeah, the Science Asylum.
Okay, I'll look that up.
I think he makes really good content, but I have to be honest, I find his presentation to be quite cringy. I mean, sorry.
Could somebody put all that in a private chat for me? Is that a channel that I should put in my stuff?
I can.
I'll do that.
Try and
Don't worry
find
about it.
Yeah, don't worry
I'll
about
find
Speed.
Hey, was Danny here earlier? Did something happen?
Yeah, he kicked me out because he couldn't take the
Oh, Danny is here.
He's still here. He isn't going to be here long.
You should read that paper. You should read that paper. That paper is exactly what I described to you. Remember in the beginning when I came in, I said there are two senses of the word mechanism in science, okay? There's the third sense is a philosophical sense of mechanism when we're talking about like robots or, the idea that, Nietzsche wasn't a mechanist in the sense that he thought that we were just robots. But the idea here is that if something is consistent in terms of parts and whole and has a function, that's a sufficient condition for that being a mechanism in biology. Okay, and so insulin fits that description. Okay, and then I said, "Excuse me, sir." And then in the second rendition, it does.
Wait, what did you say? Be nice now, Danny.
You interrupted me. You be nice. You behave, right? Just because you have power.
Fuck you.
Danny's half right.
Poor Danny. Poor Danny.
Yeah, I don't know if Danny doesn't understand what power means. I don't think. Anyway, okay, I'll read the paper. I'll read the paper and we'll discuss it on Monday's weather report, or maybe SNATA or something like that. Maybe you can come in, Danny, after you've had some rest, maybe chilled out a little bit, whatever. So anyway.
So a mechanism is not one single part. So insulin is not a mechanism, but a mechanism is an interaction of two or more different parts. So insulin is a part of a mechanism.
Yeah, I mean, even the article he linked said.
If you'd like to see the full video, head over to the Answers in Atheism channel. Link in the description. God bless.